My name is Hisham, and today I'm going to be presenting our work where we perform distributed pre-training of transformers for diverse latent representations of small molecules. In this work, we take an aggregate of publicly available SMILES datasets from GDB13, Zinc15, PubChem, as well as Campbell. We canonicalize each string in this dataset and then train a sentence piece model on this dataset. This model splits the spiral string into tokens in a data-driven manner. As a result, we end up with over 1.2 billion unique SMILES, which we perform distributed math language modeling training on. This results in our model, MFBERT. We take the results of this model, which are contextual embeddings for every token, and we use these to generate a molecular fingerprint. This fingerprint can then be used for similarity screening or as input for machine learning models. Our model can also be fine-tuned on smaller datasets to generate more targeted contextual embeddings. The architecture is based on the Roberta architecture with 12 attention heads and 12 encoder layers. We also explore additional architectures such as a Siamese MFBERT, where we clone the MFBERT network and combine two fingerprints of the same molecule but with different SMILES permutations. If you'd like to know more about our work, please check our poster P01. Thanks for listening.
Autoencoders are one of the most commonly used neural network architectures for de novo design. Their latent representations are often used to train machine learning methods that generate compounds with desired properties. However, so far reported SMILES-based and graph-based autoencoders generate latent vectors that retain information about order of atoms. Thus, two similar structures can be very far apart in the latent space. It may affect predictions of physicochemical properties based on the latent vectors. In this work, we have developed a new order-independent graph-based autoencoder architecture. Its main feature is an encoder that uses only commutative operations to transform molecular graph into a latent vector. The latent vectors of new autoencoder were benchmarked against latent vectors of SMILES-based and graph-based autoencoders, as well as ECFP fingerprints and Isida fragment descriptors on quantitative structure-activity relationship task. For this purpose, 578 subsets containing ligands tested against particular biological targets were extracted from the Campbell database version 27. Then all molecular representations were fed to a multi-layer perceptron model. The results shows that classification model based on the latent vectors of order-independent architecture performs similarly to those based on molecular descriptors and outperforms those based on the latent vectors of order-dependent architectures. Thank you for your attention, and I'm ready to answer all of your questions.
Database augmentation is an important part of training a prediction model, which mitigates such problems as small database, overfitting, and poor generalizability. Augmentation is commonly used in image recognition, in natural language, and signal processing. However, protein database augmentation has not been fully studied. In this work, we investigate the application of augmentations to a protein deleteriousness prediction upon point mutations. We augment our curated dataset by identifying top 3 unique and frequent mutations from the deleterious class and introducing the mutation to a benign class protein to increase the deleterious class samples and vice versa. This way, we make an assumption that leucine to histidine substitution is characteristic for the deleterious class, as it can only be found in the deleterious class 9 times over the dataset. Using this assumption, we find leucine in benign proteins and substitute them for histidine and change its label to deleterious to generate a new sample that belongs to the deleterious class. Our database consisted of over 3,000 benign and over 1,500 deleterious point mutations. For each entry, we had corresponding PDB crystal structures. We used baseline MCSM descriptors to extract the protein environment and sequence-based descriptors to compare the results. We trained the XGBoost model with 400 trees and 5-fold cross-validation as the training data was tabular. Sequence-based descriptors demonstrated a slight increase in area anti-curve metrics after augmentations. However, the difference between structurals and descriptors after augmentations was significant. Thank you for your attention. Don't hesitate to reach me out at the conference.
Hi, my name is Amir from Karolinska Institute, and I'll be presenting Explainable Polypharmacy Side Effect Prediction with Siamese Graph Convolutional Neural Networks. This work has utilized graph neural nets and developed a representation learning framework capable of accurately and interpretably predicting the polypharmacy side effect of any given drug combination. To achieve this, first, we established the protein side effect association by embedding them as node features of the human protein-protein interaction network. By assuming that the drug can reach the entire PPI network, and then their respective binding affinity towards each protein was calculated. Applying cutoff filtering significantly reduces the number of possible interactions. The remaining proteins are considered to be associated with recorded side effect of the drug in the CIDR database. Enrichment of this data for all available drugs will result in establishment of the protein side effect association or PSA. Next, for each drug, we extract its PSA subgraph, which contains the proteins with a high binding affinity towards corresponding drug. These subgraphs then will be used in pairs as the input of the model. The Siamese graph convolutional neural networks will predict the individual side effect, and then by mean aggregation, the model predicts the probability of occurrence of each 964 adverse side effect. The model was trained using the two sides dataset and obtains 91.43% accuracy with an external dataset containing 10,000 unique drug combinations. The pipeline is scalable and fully automated. It only requires smiles of the drugs for the prediction. Also, since the inputs are protein-protein interactions, the predictions can be affiliated to the proteins that may be causing a particular side effect. Despite the model achieving high accuracy and beating the state-of-the-art ML models, it has inferior average precision and F1 score. The reason is the intrinsic sparsity and imbalance of the data. Siamese neural nets are difficult to optimize. The model also suffers from the absence of regularization. If you are interested in this work, please reach out or visit us at posters1.
Greetings. My name is José Caetano, and along with Filipe Teixeira and Natália Cordeiro, we present our work, Machine Learning Algorithm Design for the Prediction of Isodesmic Reaction Entropy of Smaller Organic Molecules. We are scientists at the AIChem21posters1 Faculty of Sciences at the University of Porto, Portugal. Machine learning algorithms are gaining momentum as novel tools for industrial manufacturing development. The overlap of chemical catalysis and data science has proven to be a valuable asset in handling large datasets towards the understanding and prediction of the events occurring at the chemical space. With these algorithms, properties of a chemical species involved can be actually obtained, opening up new possibilities to improve quantitative catalytic reaction models. By avoiding time-consuming processes, diminishing overall computational cost, and experimental trials, it is possible to embolden chemists and industries with a predictive model that eases reaction conditions, aiming at system sustainability and efficiency. In this communication, we present our framework for setting up machine learning models able to estimate isodesmic reaction entropy at room temperature involving small organic molecules. The datasets involve experimental data from the predictive values available at the literature. We outline the advances of our low-cost in-house framework on the development of molecular descriptors used as inputs for the machine learning algorithm. The model's performance was evaluated comparing published experimental data with the obtained entropy values, asserting the pertinence of the chosen machine learning algorithms. Preliminary results show an estimated error percentage around 1%, giving a comprehensive database of isodesmic reactions, thus opening up new possibilities for using machine learning to estimate thermodynamic properties. Thank you for your time.
Hello everyone, my name is Lionel Collian. I'm working at Evotech in Toulouse in France, and I propose you to visit my poster P09 about application of Bayesian optimization in drug discovery. I will explain you what is Bayesian optimization and how we can apply it to optimize small chemical compounds. To apply Bayesian optimization on chemical structures, we need to convert structures in molecular descriptors, leading very rapidly to the problem of optimization in a high-dimensional space. In the poster, I explain how we tackle this challenge through the use of a latent space representation coming from a variational autoencoder. Then, I describe our two main applications of Bayesian optimization in our drug design projects. The first application incorporates Bayesian optimization in an active learning loop. The second application uses Bayesian optimization to generate new molecular entities optimized towards the project objective. So, if you want to understand more Bayesian optimization, please come visit my poster P09. Thank you.
Hello everybody, I'm Marcus Dablander, a DPhil student from the University of Oxford, and I'm going to talk about joint research with Thierry Hanser from LASA Limited and Renaud Lambiot and Garrett Morris from the University of Oxford. Our work is concerned with the prediction of activity cliffs in chemical space. Activity cliffs are pairs of molecular compounds which only differ by a small structural transformation at a specific site, but show a large potency difference of at least 2 orders of magnitude against a given pharmacological target. Activity cliffs are of crucial importance in drug discovery due to their role in structural compound optimization, the elucidation of structure-activity relationships, and as roadblocks for QSAR prediction models. Our key idea for the prediction of activity cliffs is to use a Siamese twin neural network that can extract features from unordered pairs of inputs in a natural and symmetric manner. Our model can be seamlessly integrated with standard molecular representation techniques such as graph neural networks or molecular fingerprints. To test our method, we conduct numerical experiments for the prediction of activity cliffs in a dataset of blood coagulation factor 10A inhibitors. We show that both a graph-based and a fingerprint-based version of our method achieve strong predictive performance and outcompete a related baseline model. In summary, we can conclude that Siamese neural networks are highly suitable models for activity cliff prediction. They are naturally adapted to process pairs of inputs in a symmetric manner, they can be easily combined with standard molecular representations, and they show strong predictive performance. Furthermore, we observe in our experiments that molecular fingerprints beat graph neural networks in our application, which calls for further research into graph-based molecular representation learning. Thank you very much for your attention.
So hello everyone, my name is Anne Goto and my poster is regarding learning from the active site, de novo drug design by combining available reagents, reactions and docking in deep reinforcement learning. So the motivation behind this work is to design de novo molecules using reinforcement learning to tackle the multi-objective optimization problem in drug discovery. And our work is based on multi-QM, which is an application of deep learning which optimizes for a reward function including QED and penalized log P. And multi-QN performs 3 actions including atom bond addition and bond removal to generate new compounds, but compound synthetic accessibility are not guaranteed. So we have used commercially available reagents from Reaxys and most commonly used reactions in medicinal chemistry from Ruffley et al. To propose new compounds. And AIChem21posters1 is a reaction-based DQ learning, and AIChem22posters1 is a hybrid DQ learning between multi-QN and AIChem22posters1, which we have developed. And based on the case study from 10 ligand pairs from the Mohalter set, it was found that our methods have improved synthetic accessibility as well as chemical space coverage. By comparing our methods against published methods, it was found that our methods performed as well or better as compared to other state-of-the-art techniques and generated compounds which are 100% chemically valid. And based on the results from Tanimoto's similarity, it was found that our method was capable of regenerating the starting compound with a score of 1 and generated compounds which are structurally similar with a score greater than 0.85. And we have further extended these methods by incorporating auto.vina, and based on the case study using one of the covalently attached COVID moonshot ligand, it was found that our method was capable of exploring active site in 3D. So if you're interested in finding out more about this exciting project to design new small molecules, please drop by to my poster, P13. Thank you for listening.
My name is Tom Hadfield. I'm a doctoral student in the Oxford Protein Informatics group. I'm going to tell you a little bit about the work we've been doing in computational fragment elaboration. So briefly, elaborating a fragment to improve its binding affinity is expensive and time-consuming. And as a result, there's been lots of recent work in looking at using deep generative models to automate this process somewhat. However, existing models for fragment elaboration either don't use any information about the target at all, or they do so implicitly by providing a list of known actives to the model. And this hinders its applicability in cases where you're working with a novel target and there are no known actives to be used. Our model extracts information directly from the protein by calculating fragment hotspot maps, which identify regions that make a disproportionately large contribution to binding affinity. Our generative model then makes elaborations such that matching pharmacores are placed within the hotspot regions. We benchmarked our model on a test set constructed from the CASF 2016 set and compared it to two recently published benchmarks, one a deep learning-based generative model and another a database-based approach. We found that, on average, our model was able to generate elaborations that were more ligand efficient than the elaborations produced by the benchmarks, illustrating the benefit of including structural information into the generative process. If you'd like to find out more about the work we've been doing, please come and talk to me at our poster session. I'm poster number 14, and I hope you enjoy it.
We proposed a model composed of a peak detection DNN coupled with a GAN for background reconstruction. The DNN model is efficient at detecting 1 or 2 peaks at most. The detected peaks can then be isolated and removed from the signal, which can then be passed to the GAN. The GAN reconstructs the area with plausible background. The signal can then be fed to the DNN again so that any remaining peaks can be detected. In the end, what we are left with is a collection of peaks and a reconstructed background. The GAN is trained on artificial data, which we prepared ourselves. To create the training data set for the GAN, the signals containing different number of peaks were generated, and subsequently holes of various sizes were made to the data. These holes are really sections of the signal that have been set to -0.1. The GAN generator then fills this area with a plausible background, while maintaining the rest of the signal intact. The GAN model we use is a Wasserstein GAN with a gradient penalty. This prevents the rapid convergence that the discriminator would have otherwise, and it keeps both the generator and the critic balanced. Both DNN and GAN have begun training and testing on real data, and while the preliminary results are promising, there is still room for performance improvement for both models.
For real-world data, we use two types of spectroscopic measurements, namely surface-enhanced Raman spectroscopy and high-performance liquid chromatography Raman. SERS data was obtained through two different techniques, substrate-based and colloidal-based. Substrate was kindly provided by our collaborators at National Taiwan University. Colloids were prepared using method described by Leopold and Lendl with the help of Applied Chem department at NYCU. HPLC data was also acquired with the help of Applied Chem department at NYCU. If you are interested in details, make sure to check poster 15.